discussion and a quick break. All right, go ahead, Chris.
Okay, perfect. Thanks so much. Hi, everyone. My name's Chris Freed. I'm a fisheries scientist at the University of California, Santa Barbara. And this is my very first EPOC event, and I think it has been just going really fabulously. So really looking forward to engaging with the community further after this. So thanks to Dylan and Riley for organizing, and thanks to Clarissa for encouraging me to submit an abstract for this. So today I'm going to be presenting a project sort of on behalf of a larger co-author group where we've been really interested in trying to develop machine learning models for predicting the contamination of California fisheries and aquaculture species with domoic acid, and using those predictions to sort of identify monitoring programs that have spatial temporal scale sort of well matched to the dynamics of domoic acid contamination in these fisheries, and also to designing sort of dynamic ocean management that is efficient in the way that it closes fisheries and reduces public health risk. So I just wanted to start off with a quick sort of simplified introduction of how domoic acid accumulates in the food web. So in the California current, diatoms in the genus Pseudonychia, under the right conditions, can produce domoic acid, which is a neurotoxin, and it can accumulate up through the pelagic food web as zooplankton graze on those phytoplankton, and then into fish as they graze on those zooplankton, and ultimately into apex predators like seabirds and marine mammals. And because this is a neurotoxin, it can have detrimental health effects and lead to sort of mortality or strandings as it gets amplified in the food web. It can also progress through the benthic food web as mussels and other bivalve filter feeders are preying on Pseudonychia diatom. And then those mussels in turn can get preyed upon by benthic invertebrates like lobster and rock crab and Dungeness crab. And then this sort of in turn affects the sort of top apex predator humans through fisheries and aquaculture. So domoic acid can accumulate in the tissue of fish and invertebrates. They're targeted in wild capture marine fisheries, and it can also accumulate in the tissues of mussels and oysters that are farmed for aquaculture. And so that presents a sort of major public health risk. So there's a lot of interest in limiting consumption of capture fisheries products and aquaculture products when they're contaminated with domoic acid. But this can lead to sort of big burdens placed on the fisheries and aquaculture industries. So right here, I'm showing sort of through space and time the open seasons for the California Dungeness crab fishery, which is the most economically important fishery in the state. So the light gray bars sort of represent the time period in which the season is supposed to be open, and the colored portions of those bars represent times when there are sort of delays to the opening of the season or early closures for different reasons. That might be due to like sort of poor body condition that leads to low yields of meat from the crabs, and here in red. But more recently and increasingly, closures due to domoic acid contamination resulting from harmful algal blooms. And there was a particularly large harmful algal bloom during the 2015-2016 fishing season, which led to really large closures, huge economic impacts, and was ultimately declared as a federal fisheries disaster, and led to the payout of millions and millions of dollars to Dungeness crab and rock crab fishermen and processors. So there's sort of a real need for developing management tools that can really effectively limit the public health risk of contamination, but can also be efficient in the way they do that to lower the impact on fisheries and aquaculture. So I just wanted to give a sort of overview of the sort of current management protocols surrounding domoic acid contamination in California aquaculture and fisheries, identify some opportunities for improvement, and then show how our predictive models might be able to guide those improvements. So on the aquaculture side, monitoring of toxin contamination is really rigorous and adaptive. So aquaculture farmers are required to submit weekly samples to the state for domoic acid testing, and are often conducting additional testing on their own. And there are very clear protocols for what to do when toxin contaminations exceed certain levels. So those actions sort of include sampling in greater areas in the farm, sampling more frequently, and ultimately shutting down different parts of the farm if contamination gets too high, and waiting to sell the product. On the capture fisheries side, things are a little bit more ad hoc, and this is going to be for the best case scenario, which is for the Dungeness crab, where we have the most sort of data-rich streams for monitoring domoic acid contamination. And that monitoring program is really piggybacking a separate monitoring program, so it's not specifically for monitoring harmful algal bloom dynamics and contamination dynamics. It's about monitoring the quality of meat in the Dungeness crabs along the coasts of Washington, Oregon, and California, to allow sort of fair competition between the states. If there's low meat condition in one state at the beginning of the season, they'll delay the opening in the rest of the states. So it's that sampling that the domoic acid contamination sampling gets added on to. And so you see this is the sort of the distribution of where survey sites are for domoic acid contamination. And before the season begins, a boat will go out, sample 6 crabs from each one of these locations, and that will trigger either the closure of an area to fishing if there are high levels of contamination, or allow that area to open on time if there are low levels of contamination. So I'm going to illustrate that really quickly for Dungeness crab. So again, we have a sort of similar plot where we see the sort of open seasons for Dungeness crab in this medium gray color, and areas in red or orange are when the California Department of Public Health declared a public health advisory, and the California Department of Fish and Wildlife shut down the Dungeness crab fishery in response to domoic acid contamination. And each one of these points represents a survey for 6 crabs, where those black points indicate at least half of the crabs, so 3 of the 6 had domoic acid contaminations above the federal limit. And when that happens, that area gets shut down, so you see it in red. And it only gets reopened once there have been 2 clean tests, 0 of 6 crabs test above the federal limit for contamination. So you can see that playing out pretty nicely in the 2015-16 season here, where the area gets closed and then reopened later once they're clean tests. So one of the but in later seasons you'll see that that the surveys will yield clean results, and then monitoring of domoic acid contamination in crabs will just stop for the rest of the season. So there are clearly sort of opportunities to improve monitoring during the late season periods for Dungeness crab. And rock crab, another commercially important invertebrate species in California, has even more sparse sampling that has resulted in really large extended closures of the rock crab fishery in Northern California, that that get reopened as soon as there is a sampling event that occurs, but the lack of sampling anytime earlier means that there's an inability to reopen that area to fishing. So there's sort of clear opportunities for these monitoring programs to be better matched in the way that they monitor the spatial and temporal dynamics of contamination events. So what we've really set out to do is try to sort of improve the ability to anticipate risk through prediction and forecasting abilities, and to use those model predictions to design monitoring programs that might better detect contamination risk. And we're able to do this by leveraging Clarissa's really amazing sea harm product, the California Harmful Algal Risk Mapping system, which provides daily gridded predictions of pseudonychia risk and of domoic acid risk in the water column from using a model that's trained on at-sea observation data and ocean color and other oceanographic variables. So we have sort of available estimates of risk of domoic acid contamination in the water column, and then through the state's monitoring program of contamination in bivalves and crustaceans that are either farmed for aquaculture or fished, we have measures of domoic acid contamination that we can relate to those domoic acid risk maps. So ultimately, we're trying to predict contamination in harvested marine species using leveraging the California Harmful Algal Risk Mapping tool. And so I just wanted to sort of go through the data that we have available on the contamination of marine fisheries and aquaculture species. This is showing the distribution of the 7 species in the study across the state in blue, their ranges, and the red dots indicate where we have samples from. So we have pretty good spatial coverage for the wild capture species, and then for the aquaculture species, bay mussel and Pacific oyster, we have samples coming from those farms. And we can take a look at sort of how frequently those samples are contaminated above the federal limit for management actions. And this gives us a little bit of intuition about which species these models are likely to work for. So Dungeness crab, we have lots of samples with concentrations above the limit. Same for rock crab, fewer for spiny lobster, lots for razor clam. But very low rates of contaminated individuals for the mussels and the oysters, which sort of immediately suggests that these models probably aren't going to work very well for them. They're going to have sort of not enough data available to classify individuals as being contaminated or uncontaminated. So the way our modeling approach works is we take the data and we divide it into a training and testing data set. We train models to predict the probability that an individual is contaminated based on its previous 30 days of domoic acid contamination risk. And we do this using three different modeling approaches, logistic regression, and then 2 more sophisticated machine learning approaches, random forests and boosted regression trees. And we do that training the models on the training data set. And then we evaluate the performance on the withheld test data set. And for the models that do well, we're able to hindcast historical contamination risk and then compare those predicted contamination risk with historical sampling and management to guide dynamic management. So to measure model performance, where sort of performance is the ability to correctly classify an individual as being either contaminated or uncontaminated, we use 3 different performance metrics. The first is % accuracy, which is useful, but it's easy to have a high % accuracy just by getting classifying all the uncontaminated individuals correctly. We look at Cohen's kappa, which is like accuracy, but it accounts for the probability of being correct by chance alone. And then we use the area under the receiver operating characteristic curve, which is shown over here. So basically, well, in the interest of time, I'm not gonna fully explain this, but it's basically the area underneath these curves. And for the capture fisheries, for Dungeness crab, rock crab, and razor clam is recreationally harvested, we see really sort of good receiver operating curves indicating like an ability to discriminate between contaminated and uncontaminated individuals. But for the aquaculture species and spiny lobster, due to sort of low numbers of samples and also low numbers of contaminated individuals, those models don't perform as well. So we're not going to really look into those further. Then this is just showing all three of those performance metrics in one plot, where we've got the area under the curve on the y-axis and Cohen's kappa on the x-axis, and the points are sized based on their accuracy. And we see pretty like good, acceptable, fair, acceptable, good, acceptable performance for models for those three capture fisheries, and then models with poor performance for the aquaculture species and spiny lobster. So again, not gonna look into this much further. So now I just wanted to look into the hindcasts of domoic acid contamination risk in Dungeness crab, rock crab, and razor clam, and sort of comment on how well historical sampling and management has been matched to those spatial temporal dynamics. And the main takeaway for Dungeness crab is going to be that the early season monitoring has been good and effective, but there's been sort of poor late season monitoring. So this is showing our model's predictions of hindcast domoic acid contamination risk in Dungeness crab over space and time. So each grid cell represents the mean estimated proportion of the crabs in a latitudinal band that would be contaminated above the federal limit. And we can assess that, and then we can see sort of tally up the number of contaminated cells over that time period and see areas that are sort of particularly high risk to domoic acid contamination. And we can compare this to historical monitoring and see how well it sort of tracks these contamination events. And you see that at the beginning of the season, it tracks contamination events fairly well, but misses some large contamination events towards the end of the season that are predicted by the model. And then we can look at historical closures and public health advisories, which are shown here, and see how well matched those were to areas of high contamination, and count up the number of sort of precautionary closures that might have occurred, or the number of overlooked closures that might have occurred, and again see that there are some large late season events that might have gone undetected.
Chris, you got about a minute left.
Thank you. So for rock crab, as I alluded to earlier, sparse sampling is sort of likely fueling many sort of overly precautionary closures. So in contrast to Dungeness crab, we see sort of briefer, shorter contamination events, and that are sort of more punctuated in time as predicted by the model, and sampling hasn't really tracked those events very well. And a razor clam has a really high, or has a really long depuration rate. It processes sort of releases the domoic acid really slowly, and shows really high contamination events. And monitoring in the sort of northern counties has been good, but at other sort of popular clamming beaches, sort of in central California, monitoring has been less good, and those areas have remained open for razor clamp collection. So I just wanted to conclude briefly by saying that these models aren't meant to be a replacement for sort of in situ monitoring and management decisions, but they can really be used to guide better sort of design of in situ monitoring used to guide those management decisions. And there's a really sort of useful feedback between improving models and improving monitoring and then improving models, and that might really unlock sort of new forecasting capabilities. So just 20 seconds. Where I sort of want to go next with this is start to do a sort of similar exercise of examining historical closures in Oregon and Washington, which I think have sort of more sort of rigorous monitoring programs, and see if we can quantify the economic benefits, the sort of return on investment in those investments in higher resolution monitoring. So thanks so much for your attention. I look forward to the discussion.
Excellent. Nice job, Chris. This was great. So with that, yeah, we're going to hold off on Q&A. I'll just go ahead and pass it to Clarissa for our next talk.